Hello.
Good morning.
Morning. How are you?
Oh, getting started. Still need my coffee.
Oh, okay. So, welcome. So this week we had, of course, our Diva Worm meeting, and in that meeting we talked about actually the Nobel Prize that was awarded for physiology of medicine, and it was given out for microRNAs, and it was two C. elegans people who won it. I mean, they worked on C. elegans in the context of winning their prize, so that was interesting. And then that was Monday, and then Tuesday and Wednesday we had two AI related Nobel Prizes. So that was interesting. It's pretty. I guess it was pretty controversial. We'll talk about that today. Um, and then Friday we had an organizational meeting on the ACT Inf symposium that's coming up. So specifically, this is for open source and open science. So we're trying to do this session in conjunction with the Active Inference Institute's upcoming symposium. This is the one on November 13th or 16th, and we are getting ready for that. So I sent out a document, I think, to Jesse and Morgan primarily, although anyone's welcome to join in. We're organizing speakers, we're organizing events. We're just trying to make this useful to people who maybe aren't as exposed to the stuff as we are, because our group has a dedicated, meeting space for that. We have programs, and we talk about it all the time. And in not in every, people don't talk about this in every group. It's just kind of a topic that we've talked about how some groups have tried to approach it, but it's well, we'll talk about that also today, about like how people think about open science. And really it's about practice, and it's about sort of the ideas, or maybe even, I don't know if there's any theory, but it's basically a lot of practice. And so that's a big part of it. And so I don't know if we're going to do any tutorials or if people are interested in that, but that might be useful. Although on the other hand, you could make the case that it's not useful, and we should just kind of give people the information, and they can, because it's not like it's particularly hard to use some of the tools that are out there. But anyways, so that's what we had this week in terms of meetings. So I guess Jesse, do you have any updates?
Excuse me. Good morning. Nothing like amazing. I didn't get to go to the Friday meeting, and I am curious about that and everything else. So I kind of missed a little bit of your recap now too. But I saw the worksheet you put together, and I'm excited to talk more about that and think more about that. On the whole, this week, it was I was basically traveling a lot this week to take care of some things, and uh, I have returned to my normal. Today is really the first day of being in my normal routine again. So I'm looking forward to kind of being able to sit down and push forward on certain things. On a side note, I'm very close to submitting a I don't really know what to call it. I guess will be a blog post on the teleology paper.
Okay.
And when I do, I wouldn't mind if you look it over to like there's some areas you could just like edit or review, and then also um, I'm I'm kind of I'm sorting out what to do next, and I think this is something that I'm going to continue to follow by happenstance, a colleague of mine is also referencing some cybernetical things recently, and we had a somewhat serendipitous talk about them, and I really want to show them the final product. So I'm almost there, and it's not. I don't think it's going to be super relevant to what they want, but more like, um, I I know this paper is something that has just a lot of ties to different things, that have, you know, it's a really interesting. The more that I read about it, look at the history of it, like it's a really interesting paper in terms of implications for all the stuff we looked at in the Varela quest line and embodiment, because the way they're situating teleology and what they call behavior relative to functionalism is really um, this thing that, one, yes, it challenges this long-standing, sort of deist, sort of purpose, theological, divine, mystical. That's a big, long-standing, arc of the the previous millennium. Is That's like a huge thing. But more specifically, the mechanization focus and the functionalism of and what causality was, and there's very small. The more I read the paper, the more I see, like, the more I understand some of Levin's, like, actual motivations in that whole branch of, like, biology or developmental or whatever theoretical takes on biology and where he's going with his ideas, but then also just the relation between an agent and its environment, and essentially like going through to um, what we call what I guess David Krakauer's, his view of complexity science and his kind of shift as taking complexity science to mean um, teleonomic matter, right? And like, why, like, I've been like this week I've been thinking a lot about, okay, like they're very clear that, you know I've been thinking about the commentary on, they explicitly say, some people say that all machines are full of purpose, or designed with a purpose. It's like, well, let's take that sentiment relative to what they're getting at, and they're really getting at the nature of purpose as being a selection of a goal by an entity, and having to get feedback from that goal. Like, if I like, and I'm really curious because there's some critiques of the paper that I want to explore, and I wonder if they get into this. But like, what do they mean by signal from the goal? And it's kind of interesting because the way they frame things, intentionally or not, is this very interesting. Um, it's a very interesting way of designating their scope, the domain of what they're concerned about, relative to, um, you know, uh, all machines. They said, all machines done on purpose because all machines aren't selecting. They aren't having to make this choice. Then they have a nice way of referencing even like a clock. And there's some things that I was thinking about in terms of the paper, in terms of like discrete versus continuous, and end states versus not having end states. Are you simply reiterating a process? Are you going forward? And at what level, like is there a way? It's fun to explore that space of, okay, well, is a clock, you know, yes, or could you argue that a clock is just constantly iterating, okay, next state, next state, next state, next state, or not? And I think there's reasons why, like, that doesn't work, and why the argument does hold. But it's really interesting to consider that relative to the way they talk about prediction, and prediction requiring multiple coordinates, one a time coordinate, one has to be a spatial coordinate, and how, like, the variation, like what they're doing in terms of associating the selection, and the selection of a goal, not just a movement, but the selection, and how the selection is tied to things in the world, that are perhaps one would say in modern day complexity science emergent, but this, it's before that. So it's just been really fun to like really continue to like. There's so much juice to squeeze from the fruit on the paper. For me anyway. I don't necessarily think that everybody loves the paper the way that I'm going into it, but like it's been really fun. It's been really rewarding to like situate um, a lot of the things we've already done with the paper. And what I'm writing so far isn't doing that. I have a couple of like at the end I'm going to have a little bit. I might even mention the Bradenburg vehicles just because it's not Bradenburg vehicles aren't teleonomic, at least in the way that they're being described here. They mentioned like they explicitly mentioned phototaxis, and Bradenburg, wrote this stuff like 40 years later. But you know, the merit of the vehicles being like really simple structures with slight changes can do these really complicated looking behaviors. And so it's an interesting juxtaposition from like functionality and how things are, the structure and how things are arranged versus all the teleonomic, all the purpose. What is purposeful behavior? Voluntary, the importance of voluntary selection in the behavior. And what does that mean? So it's kind of a nice juxtaposition there. And obviously Levin, which is like like you can, when you read the paper a few times, you can just see like, oh, here's like this is Levin like just taking out these paragraphs and making a big paper about it. It's like, okay, cool. It's like, well, if I could do that, I could do things like that too. It's like seeing some of the uh what I don't know. It's like seeing different kinds of work being done in papers is very, it's an appealing part of this. And then obviously, so Fuchs, they're, I was gonna say Levin and the Levin and Baumgart paper, but a lot of Levin always practices this thing, and now I get why. Like I understand, and even the way that the authors Rosenbluth and co did things to separate out for what, like, the more I've read the paper, the more I've understood why. Like, why did they separate out to get to prediction and higher orders of prediction? Because at first pass, it was a little bit obscure to me, like, why? But now it's like I'm getting more and more and more as to why they're doing that. And it fits really well with like a lot of Levin, what he's trying to separate out for too in certain ways. And like I get that part of it. So you have that. I mentioned David Krakauer a few times, and his like, I don't yet know what to make of his stuff. I haven't gotten further into anything from him that's on the docket. But I'm curious where he's going with what he calls quote unquote complexity science, and the teleonomic matter. And then I also put, Fuchs, who's like very much, he spent a lot of his time railing against reductionism, and has his particular take on it from the Ecology of the Brain book. So it's interesting to kind of see all those things kind of related to this paper to me. And I'm not at the end of at when I'm writing, which I'll send it to you soon.
Okay.
It's not like I'm going in depth. It's just like a couple sentences for those other authors. But like as I'm finishing the things up and like putting stuff out into this paper, it's like I'm quite curious where to go next. And I think I'm really looking forward to our meetings next week. And I'm not disappointed, but like I really wanted to attend all of the ones this week myself, but it just wasn't going to happen because there's so much stuff on my mind about it. And I think that'll be the best place to discuss it. But I want to continue on the path from here, both sort of like even maybe just kind of doing. I think maybe for either Wednesday or Thursday, whichever one we meet at, I might try to just go over this paper or this blog post that I'm writing, but then specifically kind of trace out, like almost do like expand a little bit on what I just said, connecting to the other authors and stuff, because I'm curious about that. And there's been some good links posted in both the cybernetics channel, the reading group channel, and Cognition Futures that I really haven't gotten a follow up on. And I know um, there was the one about like mechanism that I don't know if you're going to talk about that today later, to link and stuff, but I'm looking forward to that too. So that's basically the update. I'll also shout out. I think it's a little bit older, but shout out to well, what was it last week? Yeah, well, I think it was last Sunday. Shout out to Sarah for presenting at Google.
Yeah.
I meant to say that earlier, and I reposted that on LinkedIn, but I'll make like that's gonna be something really nice to celebrate. I don't know if she's gonna write a post. I know she's done a bunch of posts on Medium, but like congrats to her and everybody else too for Google Summer of Code stuff. So I wanted to say that. On a slightly different admin note, I was supposed to have a I haven't been able to engage you Albany as much as I wanted to over the last week. So I've just had a lot of things come up. So kind of unfortunate about that. But there's another event, like a research event happening at the end of this month. I might try to get us involved in some way. Um, so I'll be following up with that too. But that said, my outlook is I'm enthused because I think I've cleared up a lot of things. I'm I'm in my courses have begun, but I'm taking a lighter course load, much lighter than last semester term, which is nice. But still, there's some stuff to do. But I think the end of this year, I'm really going to have a lot of zeal to push forward on some things, and I feel good about that. So that's my update.
Good. Thank you. All right, so I have a couple of announcements regarding our Medium and our YouTube channel. So Jesse had mentioned that Sarah presented at the Google Summer of Code Contributors conference, and she presented on like a short lightning talk on her project. So it's on our YouTube channel. We have this video, The Agent-Based World of Open Source Community Sustainability. And this is, I guess, about 3.5 minutes. So it's a lightning talk. And, you check that out if you're interested in going over her project and seeing what she did, especially in the context of what we were doing with open source sustainability, or what we've been doing with it. I think it's a good one. Then she also has this demo. This was a couple weeks ago. This is the large language model powered agent-based model for open source community sustainability. This says 92 views. This has just kind of been posted. But these two videos go together if you're interested in looking at over that project. And then, of course, Sarah has also posted a number of posts on the Medium. So this is their Medium. She posted a project report on August 24, and a couple of posts like the project midpoint milestones, the building blocks, looking at the beginning of her coding period. So she has like 3 posts from the summer where she goes through the process of GSOC and then this final presentation or project report. So if you're interested in the project, if you're interested in large language models and how you can apply them to social simulations, if you're interested in open source and open source sustainability, that sort of thing is good. Okay, that's needless to say, that's going to be part of what we present to active inference. But anyways, okay, so this is the next thing I wanted to talk about. So Jesse saw this in the Cognition Futures channel, and I posted it in there, and he said, will you present on Saturday, or can we talk about it on Saturday? And I said, yes. So this is again from the Transmitter, which is a nice, I guess that's a blog of neuroscience content. So we talked about a Transmitter article last week, and this is another good Transmitter article. So They're kind of like the nature of the blogosphere or something like that. Anyways, what are mechanisms? Unpacking the term is key to progress in neuroscience. So this is by Danny Bassett and Lauren Ross. And this is, talking about mechanism, something we've talked about in our cognition futures group a lot. We've also talked about it in our cybernetics group a bit. But mechanism, of course, is something that we always talk about in neuroscience. If we want to know like how something works, we call it a mechanism. If you want to get a grant from the NIH, you better talk about mechanism. Even if you don't know what mechanism is, you better talk about it. So mechanism, of course, has this machine metaphor aspect to it, which means that a machine is a mechanism. Like a door has a latch. A door handle has, a lock has like tumblers in it, things like that. So the mechanism in the brain would be, how does memory work? And then we would look for specific cells that are involved in encoding memory and retrieving memory, or networks that retrieve memory. We might look inside those cells to look at the action potentials being generated by them, or the molecular contents. And so you can see that in a sense, mechanism is this sort of reductionist exercise where you're always trying to drill down to the fundamental mechanism, which is whatever it is that, maybe it's like flipping a switch, or it's like some simple thing that we can grasp and we can isolate and do experiments with to show this specific thing triggers, say, a memory. So there's been a lot of work with in mice on reversing memories, or reversing the encoding of memories. And people have done this with different proteins in neurons, where they've basically knocked out the protein, or they've silenced the protein, and they've been able to, basically reverse memories. Now that's a good example of a mechanism. There are other examples of mechanism, like in motor control, with different parts of the brain. And we'll talk later in the meeting about cerebellum and how, there are different cells responsible for different things. There are different fibers responsible for timing. And those are all maybe seen as mechanisms. So, in this article, they asked 9 scientists to weigh in on what mechanism means. So this is one of these articles where they try to get a consensus, if possible, to figure out what mechanism actually means. And you'd think like we'd know, but we don't. And that's kind of a problem, I think. So, the way they put this, mechanisms are often viewed as causal systems, which helps explain their central role in neuroscience. Causes are factors that control, predict, and explain their effects, giving us an understanding of why things happen and a way to target future outcomes. Identifying causal relationships in systems is necessary to understand the natural world, and the brain is no exception. And why we're interested in mechanism, of course, is uncovering the causal structure of the brain. So now, just to throw in another word that sometimes people misunderstand, we have the word causal. And of course, causal, when we look for relationships in neuroscience, we look for, relationships between things. We can have things like associations. We can have things that are correlative. We can have things that are sort of, maybe loosely related, but not tightly related. In other words, they might be in association, but they're not, if you manipulate one or if you manipulate the other, then that relationship falls apart. Now, in the case of causality, you have, there are all sorts of different ways that people have tried to figure out what causality is. One big feature of causality is this idea of intervention, where you intervene to sort of test, the causal relationship, to test it under, certain conditions. You do controlled experiments and you manipulate the variables, and you intervene and you see when those relationships hold and when they don't. We often repeat our experiments to show that causal mechanism is consistent over a large number of trials. And so that's where we get statistical significance from. And we also have counterfactuals. So we can create a situation where something occurs, and then we create a situation where it's not there. And so we have these counterfactuals that we can use to sort of strengthen the case for something being causal. So, we have mechanism, we have causality, causal mechanism, and that's sort of the gold standard of things in the brain, apparently. So, mechanism isn't just a common causal concept in neuroscience. It is often viewed as the causal concept required to understand the brain. To that end, funding agencies, including the National Science Foundation, National Institutes of Health, and top science journals, often refer to mechanism or mechanistic findings to determine what research they should fund and publish. It's clearly a status term in neuroscience, of course. And so, but we don't know exactly what they mean. So, first of all, let's think about maybe how, causality is sort of referred to in terms of the brain proper. So, standard examples of mechanisms are neuronal firing, gene expression, and antibody production, all of which fall at the molecular and cellular level. So this is where we're drilling down into like these levels of organization, as opposed to looking at, say, like a brain network or something more closely tied to behavior, and we're using this as sort of the mechanism. So the mechanism is that some gene is upregulated, a protein is produced that has an effect on a neuron, that has an effect on a network, in fact, and then it has an effect on behavior. And you can do that, but we know from like the studies, a lot of the studies of different diseases, we know from psychiatry that we can have a lot of candidate genes for something like depression or some other thing that the brain does, and it really makes it no clearer as to what the actual mechanism is or how much those genes contribute to the overall thing. So just because you use reductionism, you try to find those mechanisms, does not mean you understand the phenomenon. And then for other neuroscientists, mechanism also refers to macro-scale causal systems, such as causal circuits, topologies, and networks. So some people just say we want to get things that are at the systems level and, find mechanisms there. Those mechanisms are maybe more relevant to behavior, say. So we're interested in behavior. Everything below the sort of systems level washes out or maybe isn't as important as the causal mechanisms at the systems level. And so the systems level picture of mechanism includes higher level causal structures, which are more abstract and have spatially distant causes. So if we think about like a network, we have, maybe different parts of the network. You intervene in different parts of the network. You knock down function, you silence parietal areas or frontal areas, and it has an effect on the rest of the network. And, you can intervene there and you can establish sort of the causal mechanism of shutting down that area and its effect on the network and the behavior. But it's more abstract in the sense that we don't know all the sort of the underlying components. And in fact, those may be very different every time we do it. Not very different, but Such macro-scale systems include network and neuronal connectivity explanations of efficient signal transmission, circuit motif explorations of oscillatory and other complex behaviors, simulated lesion analysis, and brain connectivity explanations of disorders and diseases. And so what is the problem with different uses of the mechanism concept? Maybe it's just two ways to get at the same thing. But this is a problem because making progress in neuroscience becomes significantly harder when the term mechanism lacks a clear, consistent meaning. So, they offer some suggestions on how to clarify all this. In its current usage, mechanism resembles a suitcase-like word a phrase Marvin Minsky coined to describe terms that house a multitude of diverse entities. Minsky claimed this was the case with words such as emotion, love, and intelligence, each of which is a catch-all expression for a variety of things. So, a suitcase word means nothing by itself, but it holds a bunch of things inside that you have to unpack. And it's often used to conceal the complexity of a very large range of different things whose relationships we don't yet comprehend. And so, as the late Daniel Dennett stated, there is no such thing as philosophy-free science. There is only science whose philosophical baggage is taken on board without examination. So this is a quote that kind of applies here because we don't necessarily think about the philosophy of causality, and yet we use the term, and it always has different meanings, or its meanings are unclear. So that's kind of the take of the authors. Now they have different people here that they've interviewed. So this is Amy Arnston, and the question was, how is the notion of mechanism used and valued in your subfield? And talking about, this is a lab where they study the molecular bases of cognitive neuroscience, learning how molecular events influence the connectivity of higher cortical circuits that generate cognitive operations. And so kind of talking about that sort of unique challenge. Is the meaning of the term mechanism used differently in these two different cultures? The dictionary definition of mechanisms is identifying causal relationships. And from where we stand, this fundamental meaning is retained in the molecular and cognitive subfields. We see mechanisms all the way down. That's analogous to this idea of turtles all the way down, which means that you keep going down to different levels of organization, and you keep finding, you may find a mechanism, but you may find some smaller component, or you may find things that are sort of unclear, so you need to go down to a lower level still. We talked about this with the Granville paper on black boxes. So if you remember the Granville paper, where we talked about how every black box, or every white box, has two black boxes inside of it, where we start with a black box and we understand what's in the black box. So we have a white box, it's a transparent box. But even when we understand that mechanism and what's inside of it, we still have parts that are black boxes. So we still have components that we may understand how the components fit together, how they kind of operate together, but then we still have those black boxes that we have to drill down and explore some more. So it's just kind of like this endless sort of exploration of black boxes. The expansion and divergence of the molecular versus cognitive subfields has weakened our ability to understand the details of these mechanistic relationships. So, without this knowledge, research is often dismissed by reviewers and editors as not mechanistic. So, basically, what they're looking at is how mechanisms in neuroscience are often about alterations in connectivity at all different levels. Altered cognition caused by changes in the connectivity of circuits, such as correlations in functional MRI, bold signals. Altered circuit connectivity caused by changes in molecular connectivity, such as the opening of potassium channels or a synapse, weakening its effects. And the alterations in ion channels open state caused by changes in the connectivity of atoms, such as a phosphate group connecting to an ion channel rather than to an ATP. So that's kind of one take on this. Andre Fenton from New York University. How is the notion of mechanism used and valued in your subfield? This person works across subfields and kind of thinks about different ways in the domain of memory science, or learning in memory, how these terms are used across these subfields. So they, for example, combine experiments with molecular mechanisms of memory persistence and neuronal network mechanisms of spatial information processing. So mechanism in this case refers to how it works. And if you figure out how it works, it constitutes mechanistic evidence. But those two things are very different at distinct levels of biological organization. So, you have, say, for example, the ability to pinpoint structural and enzymatic changes that are crucial for initiating and maintaining long-term potentiation, elucidating these distinct molecular mechanisms of initiation and maintenance, relied on causal manipulations, employing pharmacology, pharmacogenetics, optogenetics, and genetic manipulations. So you have to use different interventions to get at that causal mechanism. It may be pharmacological, it may be optogenetic, it may be some sort of genetic manipulation. And so all those things may give you different answers, or they probe the mechanism in different ways. And so this is, but this is at this level. Now, if you look at the network level, you may use different manipulations, you have different insights. And so, unfortunately, how well causal manipulations work depends on the nature of the system, not merely the ingenuity of the experimenter. Causal manipulations may be an ineffective approach to elucidating network mechanisms, in large part because biological neuronal networks are complex systems with layers of redundancy and feedback. So this is interesting that, we're bringing in these ideas from almost, I guess they're consistent with cybernetics. We have redundancy and feedback. We have these complex systems. And, we haven't talked about that yet. We've talked about interventions. We've talked about really, drilling down and finding fundamental relationships. We've talked about networks, but we've talked about it in a way that's been kind of, trying to see, what a mechanism is. Instead of saying, well, these are complex systems with different layers of redundancy and feedback. And of course, saying something is complex on its own isn't really an answer to anything. But if you understand some of those redundancy and feedback mechanisms, they look different than, say, like a mechanism or like a fundamental mechanism that we might recognize in modern neuroscience. So if you look at gamma oscillations in memory, you can see that activity in the medial entorhinal cortex promotes slow gamma over mid-frequency gamma oscillations in part of the hippocampus, and that this activity triggers recollection. Although the electrophysiological phenomena are part of how recollection works, recollection mechanisms are likely redundant. So when you try to manipulate slow gamma oscillations, the network almost certainly compensates in an attempt to prevent a complete collapse of the biophysical origins of gamma oscillations. And gamma oscillations contribute, in fact, to more than just one recollection or more than just recollection itself. So all these things that we're trying to understand maybe have multiple roles in the system. And so when we try to intervene, maybe shut one factor down, other things come to compensate for it, so we can't really establish a mechanism. So it's a really nice insight. And so, John Krakauer, not the Krakauer from Santa Fe, but he's actually one of the people who did the cafe, Neuroscience Cafe on YouTube that we've talked about.
The other brother.
Yeah. Oh, Ed, is that?
Aren't they brothers?
I don't know. Maybe it's possible.
Oh, I thought I always assumed that they were.
No.
Maybe not. I'll check Wikipedia.
This guy has a very thick British accent, and I don't know about the Krakauer from Santa Fe, but anyways,
Gotcha.
Yeah.
Gotcha.
Anyways, John Krakauer, he works on motor control or sensory motor behavior and things like that. So basically saying that, neuroscientists really don't care about the philosophical literature, but are nevertheless convinced that proper explanations of behaviors generated by the nervous system must be mechanistic. They must show how physical parts in space and time interact to generate the whole behavior. This cogs and springs in a clock view is also expected to be causal. The clock will cease to work in predictable ways if either cog X or spring Y is messed with. So, it's this kind of like this watchmaker analogy, which of course goes back to sort of the Middle Ages and the Enlightenment, where people thought that the world was this sort of intricately placed set of entities, or intricately designed set of entities that are all in balance. And of course, that was blown away by subsequent science. But the clockwork view, maybe it kind of still persists in this idea of mechanism. And so, the idea, of course, in a watch or in a clock, is that if you remove certain parts, that you don't have, that it basically stops working in a way. So, if we think about, like, if we do a molecular study, and we knock down some molecular factor, like a protein or a gene, and we can see that, like, this affects the phenotype. There are very simple experiments you could say can do in C. elegans, where this is the case, or in bacteria. You can change the expression of some gene, or you can knock down the gene, and it will knock out the function. And you can show evidence of that. So this is the mechanism for this. Now, of course, we talked about in the last, in Andre Fenton's answer, that sometimes systems compensate for these things. So then this clockwork model doesn't really work well, right? So this is what kind of the assumption, I guess, we're using when we talk about mechanism. And so I guess that's what John Crocker was getting at. The favorite way to mess with this set of intricate springs and cogs of neural circuits these days is optogenetics. So I apologize for the blunt simplicity, but this is the stance of mainstream neuroscience. So he's making this statement that basically mainstream neuroscience is stuck in this clockwork world, or the watchmaker world. So this is where, again, it's everything is kind of there, and you either can remove a part and it doesn't work, or you remove a part and it works, and we can tell if it's mechanism or not. The idea that there can be legitimate causal explanations that are not about physical things that act upon each other locally, which would be circuits, or distantly, which would be networks, is not really taken that seriously in mainstream neuroscience. So, if we're talking about complex systems, where we're talking about, things that are, things that involve a lot of feedback, then, it's not really something we talk about a lot in neuroscience. There are some people who do, but, like, that's not the mainstream, and it's certainly not the funding agency's imperative. Okay. So let's see. Psychology and cognitive neuroscience, in contrast, employ componential functional explanations, which critically can also be causal. In an example from Daniel Dennett, which I think I either read or heard, you're walking towards a box, and I say to you, "Don't go any closer, it has poisonous snakes in it." And you come to an abrupt stop. The meaning of my utterance has made your muscles contract. This is obviously a causal story, but not one composed of a neural account of how sounds in your auditory pathway went through subsequent intermediate processing steps in your brain, culminating in motor commands. So this is like where I tell you to do, there's another person there. I tell you that this is a danger, you stop. And stopping doesn't have, it has to do with what your brain is doing, but it also has to do with the I'm hearing words, and I understand those words as being something that maybe forces me to stop. So it's a different kind of set of things going on than what we may find in individual brains. People are generating words and listening to words, and that's all in the brain. But this interaction between using language and executing actions in the world is a different kind of layer of things. And so, what is the causal mechanism here? Is it someone telling you to stop because there's danger, or is it something within the brain that is forcing you to stop? It could be both, but it's a good question, because ultimately, you could have a study where you have people saying things that are both instructional and just kind of just random, and people would just generally stop, maybe because there's danger, maybe because they're confused, and you wouldn't know if you looked at the neural mechanisms they're all the same, right? They basically, when you hear something, you stop, but the meaning is very different, and the reason you stop is very different, and that is a different a different level of explanation, and it's certainly not the same thing. Okay. So to move forward, there will need to be a broader familiarity with the more is different framework of complexity science. So here's another advocate for complexity science. And this more is different paper, something we haven't gone over, but this is sort of a foundational paper for the Santa Fe Institute. So this is a paper from like 1972 where they talk about different levels of explanation and how you can find like different granularities of explanation, different levels of explanation. So for example, you have the behavioral explanation, which is of a certain granularity. You have the neural explanation, and maybe even like the molecular explanation. Those are all different granularities, but they all kind of fit into the affective theory of why you stopped at this time. And so, this is it's actually also a foundational paper for the idea of emergent complexity. So this is the idea that you have things that happen at one level and they kind of cascade upwards to levels where you can see the effects, and of course you don't know where those effects originated. Sometimes that's not really important at lower levels where they originated, but they manifest themselves at higher levels. And so this is, he talks about how this article promotes the pluralistic idea of level-dependent affective theories. And so one might say that neuroscience is the field among those interested in the relationship between brain and behavior that is in fact defined by its requirement for mechanistic causal explanations using structural facts about neurons. So, we neuroscience is well suited for things like looking at reflexes, locomotion, and eye movements, but this can't be a suitable solution for all behaviors, and so this is something we have to keep in mind. So he doesn't think that you can, say, collapse psychology into neuroscience or vice versa. So I'm gonna, leave this up to. Actually, Luis Pessoa is a big name in sort of embodiment and activism. Why don't we look at what he's said here? He also advocates for complexity and kind of this idea that there's this Newtonian billiard ball approach to neuroscience and causal explanation that is not suitable for what we're trying to do in the brain. And so he goes again, he kind of takes the same view as Krakauer, where he talks about different mechanisms, like, say, for fear processing, where you might look at a traditional view, might focus solely on the amygdala and its molecular cascades, but this misses out on all the other things that contribute to fear processing. So, the amygdala interacts with different parts of the brain. Those interactions are modulated by neurotransmitters, and then the large scale network dynamics shift around during different fear states. So the network centered on the amygdala creates this fear sort of state, and then that state evolves over time. So that's quite a different view than, like, say, a single mechanism where you can manipulate something, take it away, or give more of it and have an effect. And so, this he kind of goes over. He has some nice views here. So I'll leave this up to you to finish reading. I'll post the article, and we can have I don't know if Jesse had anything to say about it, or Morgan. Jesse says, "Great article. We'll have to really absorb this deeply."
Sorry, you just caught me feeding the dogs here. 1 second.
All right. How about Jesse? Do you have anything to say?
Yeah. So was it only okay? So how many people Okay, okay. I'm looking at I kind of got like, so there's all these different people, there's like 10 people commenting on it.
Yeah.
Okay. We didn't go through all of them just now, do we?
No, I
I
just
just,
went through a few, and I didn't want to. I mean, I don't want to give anyone the short shrift, but like I just
yeah.
picked.
I assume some of them are, it's very interesting, kind of, like I get why they set up the article this way, but it's sort of annoying to click them open. It's like
Yeah.
I was at one of my previous jobs. I was in a situation where everybody was obsessed. It was basically what design, and
Oh, okay.
everybody at the time was just obsessed with. Geez, we got a lot of content on the page. Why don't we put it in an accordion? And just like, and then like everybody across the whole organization just was obsessed with it. And I just had the biggest flashback to that. And it's like,
Mm.
okay, like it kind of makes sense, but then it's really annoying to navigate. Um, were any of the people we looked at like, it seemed like a lot of people in this article were very like, yeah, complexity science and so on. And obviously Danny's very like, Danny's with the Santa Fe Institute. Were there people who were like staunchly against it in any way that we saw? Or like,
Well, I don't think so, but I didn't look through all of them.
yeah.
I don't think I'm in a staunch way against it. It's just that like one of the people we talked about didn't mention it, but I think it's going to be one of these things where you're trying to reconstruct or reassess causality, and so it's kind of like the way it's written is that it's well, what about causality? Let's think of different ways to deal with that, and so yeah, people are going to turn to complexity science because it's obviously the one good alternative to the sort of the view of mechanism.
it's it's really fascinating. Like I know I'm kind of I'm in a I don't know, perhaps artificially elated space, but just to really to go back to to see this in terms of the 1943 paper that we read weeks ago and I'm still writing about. Um, it's just interesting to see. Like I feel like what I want to do. Um, uh, and maybe it's because I wasn't. I don't know. I never. I don't. I'm not a trained neuroscientist. Um, so maybe I am speaking out of turn here, but like I always feel like a lot of to me this is sort of the core. Like these are like real big issues in like the nature of how science is done at large, and so much of the training like doesn't. It feels like it's something that has to be carefully unearthed, and then is So I feel like the task of um like essentially front like I never really published Frontier Map, I suppose, which I should do. But Frontier Map and the essence of that is like getting at how to connect people to these challenges. Um, and so I feel like it in terms of um, I think there's something quite exciting if you want to go there, if you want to be optimistic, I mean, which is always important to like have caveat about if you're up for optimism today about where things can go. Uh, eventually, like I think there's sort of I first I can see a certain. I'm not saying it is happening, but I can see the nature of what an inflection point would look like, where people realize, okay, we have to do X, Y, Z, and it's going to require a different approach, or it's going to require addressing the spaces that haven't been not just interdisciplinary things that built to the gap, but more like why at a large scale the processes will not account for the things that have been missed. And and the tension between that and realizing, hey, it's really hard. Like there's a reason why the easy stuff happened first. Like I think that's so important to say too, and not just demonize reductionism or demonize the mechanism. Like there's I'm against it in I guess which I'm having my quotes up, but also like why, like, it's important to understand why, this was what became established. So I mean that's really higher broad level stuff. To come back to it, it's fun. I'm going to go through the article and maybe I'll say more about it, but I feel like I think it's a great follow up from the 1943 paper. I think it's yeah, my mind is still lost in thinking, like, my mind is basically still in the mode of finishing the essay that I'm writing, whenever it is, and looking at these current works. Like now, like that could easily be put into the same space of the article that I'm writing. But then it's like, okay, well, what to do next in the space? And we don't have to discuss that here now. But it's sort of like where there's so many ways to continue to work on these topics, and it's like there's so much stuff on the table. It's like, okay, what do I pick up first? So, but that's where my mind is, and beyond that, I very much appreciate. It's a good find and a good share. When was it? Was this? How long ago was this article?
This was recently.
Oh, it was last week.
Yeah, last week.
Okay.
Yeah, it's not.
Fair enough then. That's doesn't really affect anything that I was going to say, but I don't know. There's there's a lot to follow up on it, and I just see a lot of yeah. I'll leave it at that. Thank you for sharing the article, and more ahead, more next week on these topics for sure.
No problem. Yeah, Morgan, are you in a position to?
Yes.
Okay.
Well, I appreciate the discussion. No big insights here. But I'm. Somebody was asking me what my philosophy was, and I would say that it's neural. So, yeah, and that's where I'm looking for my mechanisms,
right?
Or at least, as well as why I'm always just a little peeved with some of the kind of hammer off quantum explanation, quantum explanations for for consciousness is just like, I thought we were talking about neurons.
All right, now let me go back now. So we're, again, like I said last week or this past week, on Monday, the Nobel Prize was given for physiology of medicine, and that was a C. elegans related thing. And I talked about that in Evil Worm. And then Tuesday and Wednesday there were Nobels given on AI or AI related things, and one was in physics, which was one prize, and then there was in chemistry for protein, like for AlphaFold. So we start with this article. In Studying Nobel, when AI researchers Hopfield and Hinton take 2024 Physics Prize. And so this is, of course, for foundational work. Hopfield, of course, developed Hopfield networks, which were, these learning machines that were based on spin glasses in physics. So there's the strong physics connection there. And then Hinton, of course, developed deep learning, although I think yesterday Morgan said, I wonder what Schmidt-Huber's take is on this Nobel. Because Schmidt-Huber is, of course, famous for saying, I invented deep learning 30 years ago instead of 10. And so Hinton actually quit Google in 2023 to warn of the dangers of AI, and he was flabbergasted at the news. So this is, this is the physics prize. It was given on Tuesday. This is for the foundational work. We have a lot of foundational work, of course. We've talked about the history of that, where you have these networks. Actually, if you look at the Nobel write-up, they go back to McCullough and Pitts, which is, of course, the classic paper looking at sort of the neurophysiological logic of neurons, and that was sort of the basis for some of the early neural networks, and then, of course, that was sort of the inspiration for connectionism and cognitive science, but also Hopfield networks, which were these spin glasses or variations on spin glasses, which is a model in physics that allowed us to learn from networks, and so this is all sort of in the roots of what we might call old AI or good old fashioned AI, but it really isn't good old fashioned AI because it is more kind of connected to the modern sort of data driven AI that we use today. Well, we still use like symbolic AI, but this is the thing that everyone's hyping. So Hinton said, "I'm flabbergasted. I had no idea this would happen. I'm very surprised." And of course, this was probably, I don't know if it's very controversial to physics. I think the chemistry award was probably more surprising, at least in terms of being of a method and a computational method at that. Because usually we think of I remember like back when I was in graduate school, people would complain about like how, there was no Nobel for computer science. And apparently now there is. So it's every other Nobel, maybe not the Peace Prize, but or literature, but every other Nobel we are getting computer science in it. So Hopfield and Hinton's research, which dates back to the early 1980s, applied principles from physics to develop methods that underpin modern machine learning techniques. Their work has enabled computers to perform tasks such as image recognition and pattern completion. This one is perhaps more eye-catching because Hinton, who is often called one of the godfathers of AI, resigned from Google in 23 so he could speak freely about the potential risks from AI systems. So, as I understand it, his Nobel speech was about the dangers of AI, and some might, or I guess the threat of AI, and so some might say that there's sort of this pre-programmed Nobelitis, which is where Nobel Prize winners kind of go on these tangents. That's that's something that we can talk about later. We'll see how that plays out. But basically, why they won the physics prize is because these techniques are drawn from physics. So why are computer scientists, or I guess people in that milieu, getting a physics Nobel? And yes, Sabine Hassenfelder said, and the new 2024 Nobel Prize in Physics does not go to physics, which of course is not necessarily true because we're using sort of an applied physics point of view. So the Nobel Committee awarded this because the two men draw from statistical models used in physics, and partly because the advancements in physics research come from using the men's neural network techniques as research tools. Artificial neural networks have been used to advance research across physics topics as diverse as particle physics, material science, and astrophysics. So we're not only drawing from physics theory, but we're also enabling physics with these methods. So Hopfield is still alive, a 91-year-old theoretical biologist, with a physics background. And so the Hopfield networks are networks that describe connections between nodes as physical forces. This is, of course, the Hopfield network. And this, of course, is based on spin glasses. In particular, it stores patterns as low-energy states, allowing the system to recreate images when prompted with similar patterns. This approach mimicked dissociative memory, resembling how the brain recalls words or concepts. And this is their figure, kind of showing the relationship between natural and artificial neurons. So this is Hinton, of course, is 76 and built on Hopfield's research in the early 80s by developing a layered version of the Hopfield network that incorporated probabilities. So, this is where we're getting weights. So this is actually Hinton's work in the 80s, not for the deep learning stuff that he would do in the 2010s, but, so you have the Hopfield network that's based on spin glasses and physics theory. You then have Hinton's work, which takes the Hopfield network and incorporates weights. And so now, Hinton drew parallels to physics studies of large systems of similar elements like gas molecules. Instead of tracking individual molecules, physicists examine collective properties like pressure or temperature. So now we have this tie-in to complexity theory because, we were talking about mechanism earlier, specifically mechanism in the brain. And we can see that if we take that natural to artificial sort of analogy, we see that, like in neuroscience, we can have collective structures, emergent structures. So can we in artificial neural networks. And those are actually very important. So we don't want to necessarily track individual molecules. We can examine collective properties with these kind of tools, and the sort of link between, these artificial neurons and what we're doing in physics is kind of important. I don't know if that's something people really kind of explored philosophically or theoretically to its full extent, but it's just something to kind of put a pin in. The Boltzmann equation from the 19th century physics calculates the probability of different states in such systems. Hinton applied this concept to neural networks, naming his 1985 method the Boltzmann machine. So this is another model that Hinton developed, the Boltzmann machine. You're maybe familiar with it. This is where we have this connection between machine learning and statistical physics, which has been a fruitful area for many years. People are doing things with diffusion now, and it's a similar kind of connection that people are making. Boltzmann machine is capable of recognizing and classifying images and generating new examples based on its training data. The techniques pioneered by Hopfield and Hinton vastly simplify and approximate processes suspected to occur inside biological neural networks, like those found inside animal brains. Neural networks have brought automation techniques to machines that must deal with approximations and fuzzy edge cases. They must also learn from unstructured data. And so, this just kind of talks about, and of course it's very idealized because we know that, neural networks, it really depends on the training and the algorithm and everything, how well they do on these kind of edge cases and unstructured data sets. But that's not what this article is about. So, so then, of course, when you give a Nobel Prize, you award it to certain people. You have a rationale. There are a lot of people that kind of get the short shrift, in a sense. So that's something that, we saw with the Physiology and Medicine Prize. 2C. Elegans researchers got it. But there was a lot of concurrent work in plant biology on microRNAs where it wasn't mentioned in the prize. So that's something, where they just kind of have, they have a kind of a theme and they get people, and, some people get the short shrift. So Hinton's involvement in artificial intelligence dates back to 1972, and his achievements have significantly shaped modern generative AI. In 1987, Hinton, Rumelhart, and Williams helped bring attention to backpropagation, which is, of course, something we also are familiar with, a key method for training neural networks that is fundamental to today's generative AI models. And then, of course, in 2012, Hinton collaborated with Krzyzewski and Sutskever to develop AlexNet, a pivotal innovation in computer vision and deep learning, widely credited with launching the current era of generative AI. And so Hinton has received the Turing Award, which is the Nobel Prize at computing. This is what people were complaining about. You have the Turing Award, which isn't good enough. You'd like to have a Nobel, and now you have the Nobel as well. And so this is the other prize on Wednesday. This was for AlphaFold. And so if you're not familiar with AlphaFold, this is where we're using computational methods to solve problems in protein folding and protein structure. And so this is, of course, the computational tool. And the same sort of argument might be made that maybe we shouldn't be giving awards in chemistry for methods or for things that are tangentially related to chemistry. Of course, you could also argue that this is very fundamental to modern chemistry because you can use things like AlphaFold to predict proteins and to develop materials and things like that. So this is something that is, again, a method, but it's used, it's going to be used quite widely and it has a wide impact. So this prize was given to Dennis Sebes and John Jumper, and of course David Baker. And these are all people on this AlphaFold team. They didn't give it to the whole team because that's not the way the Nobel work, like I said. But these are John Jumper has degrees in physics and chemistry, and then Dennis has, I guess, a computer science background, a physics background as well. So you have people. Of course, people float between fields all the time, as I know, and so this is something that, again, it maybe has some controversy to it, but I think it's a good. I think it was inevitable that they gave this award to AlphaFold at some point, because AlphaFold really has improved upon the state of the art in that area. So this is about computational protein design and using this method to create sort of more efficient vaccines, speed up research on cures for cancer, or lead to completely new materials. So, of course, AlphaFold, we're familiar with how that's kind of come onto the scene. And in 2020, this is sort of, kind of took the world by storm in terms of, or at least the world of protein prediction by storm and protein folding. And so this is, of course, they've now got we're at AlphaFold 3, where we can predict the structures of DNA, RNA, and molecules like ligands. And this is, of course, essential for drug discovery. So this is something that, of course, has a wide impact in chemistry. And of course, Hessebus, and I think John Jumper was only 7 or 8 years out from his PhD after winning this. So it's kind of interesting how people can win Nobel Prizes when they're 91, and people can win them relatively early in their career. So, and then of course, Baker works more generally on protein prediction. So Baker's created several AI tools for designing and predicting the structure of proteins, such as a family of programs called Rosetta. In 2022, his lab created an open source AI tool called Protein MPNN, which is able to help researchers discover previously unknown proteins and design entirely new ones. So it's actually maybe more for a class of tools that are being developed, chief among them AlphaFold. So this is the other prize. And so if we go here, this is the nobelprize.org site, where they kind of show kind of the profile of the people. They always have the serious drawing here. And then they kind of describe why they won the prize, and then have some, like a press release and some other scientific background. And then this is the prize in chemistry for 2024. And, press release and scientific background as well. So that's what that's all about. Now, I mentioned that, why is it that we're giving a Nobel Prize to a method? Both of these are kind of method oriented. So, there's some fundamental physics here. There's actually some fundamental chemistry here. Well, why would you give a Nobel to a method? And so people have asked that question. And all you have to do is go back about 31 years. So in 1993, Jurassic Park came out in the theaters in the summer, and then in the fall, we get the Nobel Prize for PCR, or polymerase chain reaction. And so this was won by Kary Mullis and Michael Smith. This is definitely chemistry, but it's a method. It's not like for some fundamental discovery. It is a method, but the method is wide-ranging impact. And, you can do a lot of things with PCR as much as you probably will be able to do with AlphaFold. And so this is This was the Nobel Prize in chemistry for 1993, awarded for contributions to the developments of methods within DNA-based chemistry. Awarded to Kary Mullis and Michael Smith. Kary Mullis did this sort of invention of PCR, or at least kind of perfected it so it worked. So PCR, in brief, you have a template of DNA and you can put it into the set of chemical reactions, and it's this what they call thermal sequencing. So like you have this template of DNA, and you basically subject it to these heating and cooling cycles, and you can amplify that DNA through these cycles. So you program a machine to go from like 95 degrees down to about 10 degrees Celsius. You get this cycle, and it allows you to replicate DNA or RNA sometimes in your sample. So this has been very useful for all sorts of sequencing tasks. That sort of forms the core of next gen sequencing, and a lot of techniques for analyzing DNA and RNA. So this is important for genetic engineering. It's important for genetic analysis, all of that. And then, of course, Michael Smith was able to do something called site-directed mutagenesis, which was, of course, a technique to do these targeted studies where you can target certain oligonucleotides and have effects in the organism. So these kind of went together in this prize. And then there was this article I found. So there's this article I found on, it was kind of like, I guess, tongue in cheek, but it does sort of continue from this sort of AlphaFold and PCR tradition, which is, when will BLAST get its Nobel Prize? And so BLAST, of course, is a bioinformatics tool for searching for gene and protein sequences. Given a template, you do this computationally, and you find the best matches in a database to the sequence that you're querying. So this is actually kind of tongue-in-cheek, but basically, people have complained about how you're awarding Nobel Prizes, the methods. This person says, I think the creator of BLAST should get a Nobel Prize. The impact that BLAST has had on the field of biology, not just on bioinformatics, has been huge. These people created a verb. What modern biologist does not know what blasting a sequence means? The BLAST paper is one of the most highly cited papers in history. Its impact on physiology is undeniable. Lippmann and Gene Myers, who are the people who developed BLAST, stand out for their contributions to the computational processing of biological sequences. That's the Nobel sort of description of BLAST. I know that BLAST was built on previous work like FASTA, but one, so is everything, and two, FASTA is also Lippmann's work, so he can claim credit for that. The other counter argument I've heard is that BLAST is mostly a method, but so is GFP, which is green fluorescent protein. And one may argue, and of course this is one, I don't know if this won a Nobel, I think it did. One may argue that it was very cool that one could have a protein that fluoresced by itself, but the prize was awarded for the impact in the lab. Does anyone believe that just the 1962 discovery of jellyfish protein would have sufficed for a Nobel? So this is where, let's see. Yeah, this shows in 2008, this was the award for glowing proteins. So green fluorescent protein is a subset of all the kinds of colored fluorescent proteins that we have. And we use these ubiquitously in biology to tag things. So we use like some sort of, we use it as a tag to, attach itself to some molecular target. And then we use it to report on whether that's present or absent in a sample. So this is a very useful tool, but it actually comes from jellyfish protein. It was isolated by a Japanese researcher, and then they were able to purify it and give some value. So Marty Chalfie, who's a C. elegans person, was able to use GFP as a genetic tag for different biological phenomena. And then Robert C.N., who's a big biochemist name in biochemistry, was able to help us understand how GFP fluoresces. So that's what they're referring to there. And so, you could argue that you could give Blast the Nobel Prize, but, this is tongue in cheek, but still. And then finally, this article from John Hopfield in October 2018. This is after, I think, winning some prize, and then, not the Nobel, but basically asking now what. So, this Hopefield, of course, is 91 now, so kind of just, basically retiring from his position, winning the 2019 Franklin Medal in Physics, and kind of going over some reflections on what physics is. So to me, growing up with a father and mother, both of whom were physicists, physics was not just subject matter. All things in the world are incidentally the subject matter. The central idea was that the world is understandable, that you should be able to take anything apart, understand the relationships between its constituents, do experiments, and on that basis be able to develop a quantitative understanding of its behavior. Physics is the point of view that the world around us, with effort, ingenuity, and adequate resources, understandable in a predictive and reasonably quantitative fashion. Being a physicist is a dedication to a quest for this kind of understanding. And so kind of reflecting on his life and some of these things. And so this is kind of an interesting article if you're interested in, some life of a scientist who ends up winning a Nobel Prize. So that's kind of all that was about this week. Any comments or questions on that?
Yeah, I mean, it's if AI begins to deliver what it's promised, we should see more of this. Yeah. Or like more will come of software and software computational innovations.
Right.
I wasn't. I'm hoping that we do something at Celsius that's around computational protein design, just in terms of some appreciation of that work.
Yeah.
And maybe AlphaFold too. I mean, certainly, the big thing that the AlphaFold team could objectively stand behind was the decades-long protein structure competition. Right. That they certainly had their AlexNet moment in terms of just smashing all previous records in their first release, right? and even I believe improving significantly on the next two. So there was a lot of just wow, what have they done? The thing we're not talking about is the fact that like now I don't want to say people have moved the goalposts, but it's just like maybe protein structure wasn't the great wall holding back biological innovation.
Yeah.
And yeah. And I don't know if you mentioned Ben Blazek's comments about this is possible because of lots of data that has been stored in databases you know, like you said, like I think you posted something after Ben's thing where it was just like, when will the protein database get its Nobel?
Oh yeah, that was the thing I talked about.
Yeah, and just something, you know, again, something to be said for, you know, that I mean, it always seems kind of obvious that language models would be the first, you know, in quotes, breakthrough,
right?
Just because of the data, right? And you've seen what the companies have done, or, you know, like from like trying to pay people to generate more text to, you know, finding weird text stores that they could buy access to, but you know, at a certain level, there's been a plateau in performance because it's like we've finished all the data,
right?
And in biology, I mean, I should have posted this too, but I posted the UK Biobank scientific conference videos that dropped this week.
Okay.
I think from last year, you know, like they're not super recent, but they only just made it to YouTube. And it was interesting to see Daphne Kohler, who's a famous Stanford machine learning professor, who I believe is now, I don't think she's kept her Stanford, you know, she's joined, like a lot of Stanford machine learning professors, you know, she's gone into industry and got you know, a huge amount of VC money to start a biology company.
Yeah.
And it was interesting because I had just posted something of her speaking, I think, at the World's, what's that? I've never understood what that conference is. It's like the World Scientific Forum or something like that.
Maybe.
Seems very splashy, but it doesn't seem like it's a scientific conference. Anyway, and she was speaking in kind of big generalizations. What was interesting to see her presenting at the UK Biobank conference, because there it was just like, oh, you know, she's not just one of these, like, I'm trying to do a DNA foundation model. You know, she's discussing like the histopathology and multimodal data that's associated with the it's associated with the UK Biobank, you know, and the future of bio, you know, the one UK Biobank like data sets don't really exist anywhere else.
Yeah.
Right. It is an amazing resource. And two, as amazing as that is, how it's still, you know, it is still just one data set. And you know, you are kind of limited to the tests that those people did, and it would be hard to add to them, right? Like it's physically hard. It would be physically hard to say like, oh, I want to just track down that same, you know, 1 million UK people and do this thing to them, right? Like this isn't software, and like each one of those data points is potentially expensive and or hard to do, in the sense that like doing those measurements requires potentially a you know, a skilled technician.
Yeah.
And that biology perhaps needs, like, you know, it needs like internet scale more of UK biobanks, right?
Yeah.
You know, yeah. Just because, again, you know, the biology of the mechanisms to tie it back to the mechanisms of life are a lot more complex than the syntax of language, right? And look at the you know the ratio that we had in terms of data to algorithms.
Yeah.
You know, quote mechanism in terms of deep learn, you know, in terms of training neural nets to be chat GPT, right?
Right.
It's anyway.
Yeah, interesting stuff. We'll see how in the future if we get more Nobels on AI. Certainly going to have a lot of applications though, and but that's been going on. And it should be said that the prize in chemistry, you know, we talk about AlphaFold sort of in isolation, but really it's based on what people have been doing in chemistry for years. And, you know, is it like, you know, it's sort of a continuation of that. So it's like as you're getting more computer power, you can solve these problems more. And I like your point about how maybe protein folding wasn't the barrier that we thought it was. Just simply you get enough computational power and you write the right program, and it's not that hard because you have the, you know, it's like any other set of problems, like where we thought it was really hard, and it's, you know, when we get enough computational power, it's not as hard as we thought, not as intractable as we thought. So.
And it kind of it does set our sights on the next problem, but in this case, it's actually problems, right? In the sense that it's not just docking, but it's also that there are plenty of that protein shape is actually you know somewhat probabilistic,
right?
You know, you're potentially multiple shapes,
right? Just anything that's conformationally stable. It's like
yeah, and so docking has a number of interesting problems to it, and all the other protein interactions. Is Hinton on the all you need is attention paper?
I'm not sure. I don't think so.
I don't think so, right?
I don't know.
I mean, you know, it's again, just it's kind of funny that we're all, you know, or at least right now in this particular hype cycle, it's the I posted a thing on the or one of those culture papers is sorry, it's attention is all you need.
Yeah.
Right?
Yep. Who are the authors on that? I don't know if they're any.
It's a bunch of Google people I don't recognize. Okay. I think it's all Google people, right? No, it's got a University Toronto. Yeah, Aiden Gomez, I think, is one of the like these are the people who went on to. You know, I bet you these are all people at startups right now.
Yeah.
You know, and when I say startups, I mean, you know, that have more than like $100 million.
Right.
Like, not startups.
Yeah.
You know. But Google Brain, Google Research, and University of Toronto, and then last author Ilya Poloshukin.
Okay.
But I posted a link to the video on like RNN's all we really needed or something like that.
Right. Yeah. Yeah, that was a kind of a presumptuous title, as it turns out.
Well, it's certainly been riffed on from then on.
Yeah.
Everybody's substituted there. Yeah.
So yeah, I'm actually a member of the ASAP Bio Slack. So ASAP Bio is an org that does a lot of stuff with open science, mainly with preprints, but other things. Basically an advocacy group, and they've been doing a lot of interesting stuff with um, different types of events. So when I was part of ASAP Bio as one of their cohorts, we hosted this talk on the past, present, and future preprints. We put together this talk where we had a bunch of guest speakers talking about their experience with preprints and how they use them in their labs. So this is a very good presentation. I actually live tweeted this at the time, so it's very much it was during the pandemic, so it was very much something that we had to do virtually. But it was a good experience. Anyways, this is what they're doing. That's what they kind of advocate for, and so every year they kind of come up with these sort of focal areas of their efforts. So I thought this was good because it kind of gives you an idea of the lay of the landscape in open science and what people are thinking about in terms of maybe the types of things that people want to do, or kind of the open questions. So this is it's a community manager, Johnny Coates. 2025 is rapidly approaching. And we're now planning our community calls for next year. So they do these community calls where they work on these projects and kind of work on something having to do with these questions. So as with last year, I wanted to give you all an opportunity to vote on which calls are held so that we're delivering things you want to hear about. So these are things that they kind of identified as major themes in the world of open science, kind of open questions, but also things you can do project on. So, like the first one has to do with preprints and publications, predatory publishing, paper mills, and their impact on scientific publication and preprints. So this is where people try to game the system, or they write papers with generative AI, or they kind of write derivative papers, and there's this sort of predatory aspect to publishing, or this later sort of mass production of published papers that's kind of a problem in a lot of not only in journals but in preprints as well. So that's one, and that's something that's kind of not directly related to open science, but it is an issue that you can have an open science angle on. The second is institutional recognition of preprints. So in open science, there's this issue of practice, where you want to impart the practices to people so they can do better science. And the assumption is that if you do open science, and that the assumption is that's better. But it costs people time, you have to implement those practices. But moreover, your home institution has to recognize the value of preprints. So if you publish something as a preprint, does it get viewed as a publication, or do people just dismiss it? If people release preprints early, do they get, recognition for it? Or, what is the role? How do people view preprints? Some institutions view them favorably, some unfavorably, some are, sort of neutral. And so this has a role sometimes in tenure and promotions. This has a role in sort of, as we know, like in the world of AI, preprints are ubiquitous. Everyone posts their work on arXiv. But in some fields, like in biology, it's a mixed bag. And so that's kind of what they're getting at there. There's also publishing experiments. We have Arcadia, which is a company that was founded to do like exploratory science, but also built on different new publishing models. So they're doing like replications, they're doing other types of working with different types of model organisms. and then pushing the work out in these innovative types of publications. There are other platforms like PubPub and other types of like preprint aggregators, and it's there are all sorts of experiments going on in the publishing sphere. And, that's something that I just got an inquiry from the Active Inference Institute about setting up a overlay journal. And overlay journals, of course, are where you host some papers. You might have like a thematic type of journal atmosphere. You have an interface that basically presents papers as like being part of a journal. You have editors, but you host your papers on, say, something like Open Science Framework. So you can actually build a journal from make it very easy to build a journal and sustain it. So this is something we're looking into. So that's that. And then social media and science communication, of course, having the outreach aspect, but also having the communication aspect. So rapidly reporting results as sort of blog posts or, social media posts instead of you might write the paper or the preprint, but then you have to get the word out. And this is where social media and science communication kind of come together. Also, we have science communication on social media. We want to sort of interpret studies for people, make sure that they don't fall prey to misinformation, or that, there aren't like sort of misinterpretations of an article. I'm sure you've seen things where there are very sensational headlines about something, but of course, those sensational headlines have to be countered because they can be very damaging to really understanding what was done in the study. Journalists and preprints, responsible reporting. This is kind of what I was talking about, where someone who doesn't have the domain expertise picks up a preprint, says that this says something that it doesn't, and then that kind of persists in the minds of people who are kind of casual, have a casual knowledge or maybe no knowledge at all of a certain area. So, when, this was particularly important during the pandemic when you had a lot of preprints that were, not, shall we say, not necessarily about the science. Anyways, this is a constant problem, especially when universities release press releases on articles and they don't necessarily match what the actual work that was done. It doesn't match that. So this is something that, it's an interesting topic. And then, of course, there's this topic, routes to open. Where do preprints fit within other OA initiatives? So how do you get to an open science practice, or how do you get to open science practice? You might do this through preprints. You might do this through science communication. You might do this through sharing data. You might use all three as a strategy. You might have like videos, like, our lab, we do a lot of videos. We do a lot of papers, blog posts. We have a very varied set of channels that we use, and some people are just satisfied with maybe releasing preprints or releasing open data. And so, they're interested mainly in preprints, but it's kind of interesting how those kind of strategies intersect. This is actually, the next one is a really interesting question, and that's, has open access failed? So, maybe, we could ask the question, how widely adopted is open access, sort of open science practices? But of course, journals have this imperative of open access where, instead of having things locked behind a paywall, you have access to them. And so if you go to something like PLOS or eLife, they have a model where the author pays money to have the article be freely open. And sometimes those fees are extremely high. And it's hard for researchers who don't have a lot of grant money or a lot of institutional support to pay that fee to make the article open. There are different types of access. There's green open access, diamond open access, gold open access, and they're basically just variations on how to make something open access. But there's this issue of, kind of where companies, the big publishing companies like Springer and Wiley have kind of unscrupulously taken the model and used it to kind of make a profit. So they're squeezing authors for money, and they're also squeezing libraries, academic libraries, for subscriptions. And so, has open access really kind of facilitated access to articles, facilitated access to open science, or is it just another money-making scheme? And that's an interesting question. I feel like kind of it's been co-opted in a lot of ways, unfortunately. But it's something that we have to think about. And of course, with overlay journals, we can get around some of the costs associated with running a journal. So the whole reason why, we're reliant on big journals, well, because there's a prestige aspect, but there's also an aspect of it's hard to put together a journal with editors and with reviewers, and then with the infrastructure to publish something. But now with preprint servers, we don't necessarily have that problem. We can take a preprint server. We have other tools that sort of enable post preprint review, or something like that, and we can put together a system that approximates a journal. We can also do this with overlay journals, where we can host the articles with a sort of a persistent identifier, and then we can have an interface where we can allow for reviewing, comments, displaying all of the supplemental data and everything else. And then finally, this is a sort of a evergreen topic, which is mis and disinformation in preprints. So like when people kind of misinterpret their data, or there's like basically a bad faith preprint that comes out arguing for something like, some drug being used to cure COVID or something like that. Those are things that we want to sort of identify and deal with as a problem in the community. So those are kind of their open questions. I would probably come up with a different list, but this is what they're interested in. So I have a couple of articles here. This is the first article. This is an interesting article from Frontiers in Neuroinformatics. This is open is not enough. Let's take the next step. An integrated community-driven computing platform for neuroscience. So this is a paper that I think we talked about this yesterday. And those are
the NeuroDebian developers.
Yeah, this is the NeuroDebian.
Okay.
So some of these are going to be about like our conversation yesterday. And so this is a paper that kind of describes sort of this NeuroDebian platform, which argues that, we have this sort of standard of open, and, just making things open isn't enough. You just don't throw your data out on the internet or put out preprints and, really have no, it can be sort of divorced from the community, and that you're just putting things out in the world without any sort of community feedback or community involvement, or not letting the community learn the practices and use them easily. So this is what they're talking about here. And this, has connections to INCF. So, they're trying to kind of get a handle on a lot of the neuroscience software projects that exist. So in scientific software, we have two problems. We have sort of this model of sort of homebrewing everything, which is like where you create your own software for, doing something. And then you make it open. But of course, making that open doesn't really solve anything, because you're not going to maintain the software, and you're not going to, make it easy for people to use. So making it open doesn't really make much sense. So there's been this movement to have like research software engineers, where labs would hire a research software engineer who would make things very much like a more professional software project. But that, of course, costs money, like most open science initiatives. So this is a problem. And of course, making this sustainable, that's another issue. So, we know that software research, software projects, that whole culture is broken. Inefficient and opaque procedures combined with a scarce developer workforce results in tools of insufficient quality and robustness. So we need things to conduct the research, but we don't have those tools, even if they're open, because we can't use them. And people, of course, leave their positions, and when they leave their positions, usually the software dies with them. So we need to bring our tools into the open. We need community-driven platforms so the entire community can contribute to these different tools. So this is where we talk about NeuroDebian. NeuroDebian applies proven principles and procedures from free and open source software, or FOSS, and this allows us to sort of develop a strategy. So NeuroDebian strategies to work with scientists and developers, helping them to directly integrate their software with the Debian operating system. So using this, Linux distribution, we can integrate the software into that, and then people can use that, and it's all open, and it doesn't cost the labs very much to implement. So they're trying to sell NeuroDebian here, and Debian in particular. There's of course NeuroFedora, which is a different platform, and of course Morgan is very familiar with that. And
yeah, I started it because of these guys.
Oh, okay, yeah. So yeah, they talk about having strict open standards. So the other problem, of course, in scientific software development is that we don't have good standards, and this is true for open data as well. That we need to have standards that people adhere to. If we're going to throw things out into the world, you can make things kind of just throw them out there, and if they don't follow standards, if you don't have good practices that are allow for replication, then the things that you put in the world aren't useful, particularly useful. So this is where they have open standards, where they want maximum interoperability of all components without sacrificing robustness or creating unnecessary burdens. So this is, they kind of go through all of this. They talk about how widely used NeuroDebian is around the world, and it's of course most of the industrialized world, with some, well, it's pretty worldwide, I think, distribution-wise. And so it's pretty representative of the world. And, we have these, so not only is it built for widespread use and replication and sort of this professional standard of software development, but it's used all around the world. So this is one paper where they kind of advocate for certain open standards in science. And then this is an article more on reimagining peer review. So this is a different issue in open science. This is where we need to kind of rethink peer review, because you can have, usually the way peer review works is you might have 2 or 3 reviewers on a paper, and those reviewers might have different abilities to sort of scrutinize your paper. And so you make it through those hurdles and you get published. And of course, as we know from a lot of the studies that have been retracted, sometimes you can commit out and out fraud by manufacturing data or manufacturing figures. And you can get through those hurdles pretty easily and still get your paper published, sometimes in really reputable journals. And so the whole idea of peer review is that you're kind of trying to be held accountable by peers. But the problem, of course, is because you only use a very small number of peers, that that can be, sometimes it can be a barrier where your peers don't want something published, or where they have a beef about some theory or something. And so it can be a problem there, but also that you only have 3 eyes on it, and they usually don't say scrutinize the data, the numeric data, or they don't think too much about the figures. And so this could be a problem where you actually don't get the desired result. So people want to reimagine peer review. There's been crowdsourcing, there's been other types of peer review, open comments on papers. But when we do this, we also need institutional buy-in. And so this article is reimagining peer review needs publishers and institutions that collaborate. So the abstract of this paper reads, collaboration between academic institutions and publishers is essential for advancing ongoing peer review reform. Despite being an important process in scientific publishing, the flaws of the current models of peer review, used most by publishers, which are usually the model where you have an editor and you have a couple of reviewers, and the editor will send it out to reviewers. The reviewers will give their reports back. The editor then makes a decision, and then, it's either kind of driven by the editor or driven by the reviewers. But in both cases, you have people who maybe don't understand the paper fully, or maybe have a bias, or maybe don't scrutinize the paper at the level that maybe it's scrutinized at later. So like the numeric data or, details of the figures. And there are things that, it's human error, it's collective error, it's that you're not sampling enough from your peers or something like that. So there are a lot of flaws actually in peer review, but the flaws of the current models of peer review used by most publishers are increasingly recognized and include inefficiency, inconsistency, bias, and a lack of transparency. So these are the things that they've identified. Fortuitously, numerous journals and related organizations have leveraged the transformative potential of preprints to already initiate positive changes. However, active support for academic institutions or active support from academic institutions influential in shaping researchers' careers and cultures is crucial too. This potential collaboration would offer mutual benefits, foster more responsible research assessment, help reimagine peer review, and ultimately promote a healthier research culture. So in this case, we're talking about institutional buy-in from people doing open science, just not only the scientists and the peers who are reviewing it, but the institutions as well. Any comments or questions?
Yeah, peer reviews. It's like one. It's almost like a deeper social, not problem, but it's also managing the I think we talked about on Friday. It's like managing the expectations of what comes from peer review.
Yeah.
And managing the difference in expectations that comes from peer review, because different people have different ideas, right?
Yeah.
And then there's and the many different problems that lie behind reviewer 2.
Oh yeah. Or reviewer 3. Yeah.
Or yeah. Yeah.
I mean, you also don't want to make it a free for all, so that people can be.
You don't, it's but like we said, it's not exactly, it's not designed, nor perhaps even appropriate to treat it as a fraud detection.
Right. Right.
It's a it's a review, but it's in a hopefully collaborative kind of way, right?
Right.
It is hard for us to not treat it like it's your grading papers.
Yeah.
And it may be because we're asking academics to multitask so much, and grading papers is a big part of things.
Yeah.
humans are other than computers. It's hard to switch off certain things that you're spending a lot of your time doing.
Yeah.
But yeah, it's just sort of to follow up. I think I posted this, but I don't know if you saw it. That Guillaume, the previous lead developer of SPM, which is the kind of certainly one of the premier tools in functional neuroimaging. And a MATLAB package that goes back to the 90s, and is basically Carl Friston's. Carl Friston started it, and it is the repository for his many computational contributions to statistical parametric mapping, right? Which is the name of the tool itself, right? He's moved on.
Yeah.
Right, like I see that, like last year, it's only been like a year he's moved. So I did ask him if he's I asked if he's still working on Octave at all as an open source developer, and if he's still interested in giving a talk in that kind of speaking to that kind of work, but he might be lost to industry.
Yeah.
And then just thinking about I'm just trying to remember what his okay, it is the Center for Open Neuroscience.
Okay. Yeah.
Yeah, so I kept on thinking Center for Science, which of course is the Virginia thing, but it's Center for Open Neuroscience.org. So he would be a good speaker.
Yeah.
And he definitely has talks on the subject.
Yeah.
As you can see, like I knew him, or I had email contact with him when he was still a grad student in Rutgers, and his advisor was that neuroimager that was quite involved in kind of early neural network work. I'm blanking on his name right now, but Steve something. But like good early, early 90s neural network work. Did you have more papers?
Let's see. So this one here, we'll talk about this. This is a paper that Ricard Soleil and Michael Levin are on. This is called Open Problems in Synthetic Multicellularity. And it kind of talks about some of the things you were talking about with respect to neuroscience. This is a concept paper that they put out online recently. So yeah, this is Open Problems in Synthetic Multicellularity. So the abstract of this is multicellularity is one of the major evolutionary transitions. So, we think about evolutionary transitions, what kind of has led to a lot of the biocomplexity that we see in the world today. One of those is single cells living, alone, and then single cells living in colonies, and then single cells living in these multicellular communities where, they resemble tissues which are functionally interdependent. And, so you can have single cells kind of living their lives in the world. You can have these single cell collectives where you have different colonies. And then you have this multicellularity, which is where cells become interdependent in tissue or something like that. There's this evolutionary transition. You see those intermediate steps still around in organisms like yeast. but this idea that you have this evolutionary transition where you go from cells being sort of self-reliant to cells being in this community. So the rise of multicellularity provided the ingredients for the emergence of a biosphere inhabited by complex organisms. Over the last decades, the potential for bioengineering multicellular systems has been instrumental in interrogating nature and exploring novel paths to regeneration and disease, as well as cognition and behavior. So this is kind of where they're talking about these sort of, this is much like what Mike Levin does with, cell sort of synthetic systems, and then also some of his work on tame, because what they're going to be doing is engineering cell collectives that have certain competencies, cognitive competencies. And then, of course, Ricard Soleil is interested in sort of the complexity of that and how, we got there through evolution and how that complexity is managed. So this is kind of, they kind of go over a list of open problems, ongoing and future challenges in the field, and conceptual approaches that may facilitate progress. So this is kind of a sort of a taxonomy of natural and synthetic multicellularity. So you have natural multicellularity. You have this governed by a number of different mechanisms. So again, the word mechanisms. You have hierarchical mechanisms, and that's something you see in the embryo. In this case, this is a Drosophila embryo where you get striping, you get different pattern formation. This is governed by hierarchical gene expression, and we have this kind of where the cells sort of work collectively to form these patterns in the embryo. You also have emergent mechanisms. So this is an embryo of a vertebrate where you have this emergent mechanisms where the tissues are formed through this emergent complexity. And then you have evolved complexity. So this is, I guess, a group of multicellular cells. This is evolved cell assemblies emerging under synthetic selection mechanisms. So this is where we can do sort of artificial evolution or simulated evolution experiments where you get these ability to form these multicellular complexes. So those are natural examples. Then we have synthetic examples where we have, again, the hierarchical mechanisms, the emergent mechanisms, and the evolved complexity. So in B, we have a synthetic bandpass filter using engineered E. coli. C is, let's see. I don't know. Okay, I think they labeled B twice. But I think this is C, the generation of multiple coexisting cell fates. In this diagram, you can see the different colors. D is programmable symmetry breaking into structure formation. So this is where we have basically structures coming from where you can program symmetry breaking between cells. They form different tissues and you get this differentiation within this mass. And then morphogenetic processes are spatially organized through multi-scale feedback loops shaping embryos. And this is where we talked about F, G, and H. So these are the emergent mechanism examples. F is a kidney organoid development example here. You can see the organoid. It's not, it's basically a bunch of kidney cells, kidney-like cells in this organoid. G is Turing-like branching morphogenesis of bacteria. So you can see in the culture here, you see this branching coming out from a central core. This is a bacterial colony. It's showing the sort of Turing reaction diffusion pattern formation mechanism. And then H is the development of anthrobots. So this is one of like a xenobot. I guess it's an anthrobot. I don't know what the difference is. Maybe they're using human cells for this instead of xenopus cells. But basically, this is one of their one of their examples.
Epithelial cells.
Epithelial cells. Yeah. So this is where you're creating this hybrid robot, but with cells from a cell culture. Then evolved complexity is J, K, and L. J is where are we? Yeah, they didn't label their figures as well as I could have here. I guess this is basically so evolved cell assemblies emerge under synthetic selection mechanisms, including cell-cell adhesion to escape from predators. So J is the cell-cell adhesion example. K is adapted from this reference 8. And then xenobots are here. So this is a xenobot. So you have these different versions of sort of mechanisms and ways to get to this complexity. And then you have examples from the natural world and a lot of examples from the synthetic world. Now, when they say synthetic, they mean like under controlled conditions. So like a bacterial colony is not really synthetic in the sense, I guess you can program the genome, but like, some of these are computational, some of these are sort of hybrids, some of these are organoids, and some of them are cell culture. But basically, it gives you, I think, a good overview of what we can do biologically and what occurs in nature. So this is, the current landscape of synthetic multicellular systems can be roughly decomposed into 3 partially overlapping classes. So this is where they get into sort of the structure of the field and sort of the challenges that we have to sort of deal with. So the first is synthetic multicellular circuits. And so this class involves cellular circuits that have been modified or introduced through genetic engineering with living cells, typically used as a chassis. So this is like your xenobots or your anthrobots. And then many designs within this domain rely on a modular approach to circuit complexity based on standard combinatorial circuit design. Cellular consortia have been used as a multicellular implementation of all kinds of simple responses, from combining Boolean gates to pattern formation. These designs involve strains interacting through chemical signals, propagating in a liquid medium, or diffusing over short distances on an agar plate. So you can build these synthetic multicellular circuits, which are gene circuits that do certain things, express certain products, or they actually can fit together to give you this great circuit complexity that can perform things like computational functions like Boolean gates or involve pattern formation. And that's something you can design. So like a lot of your bacterial example of bacterial pattern formation that we saw there was this sort of thing. We also have programmable synthetic assemblies. The next step towards engineering multicellular systems is exploit the predictable properties displayed by adhesion-driven spatial morphodynamics. So this is where we had these these cells that were joined by adhesion, and they're distributed in space, and we can sculpt those kinds of assemblies in a very profound way given our interventions. Again, this bottom-up engineering allows predicting or programming the outcome of the final spatial structure. It was easily understood that cell sorting due to a different adhesion energy, or cell sorting due to different adhesion energies, could easily explain self-organized aggregation of a set of randomly mixed cells. So, when they did early cell culture experiments with cells that kind of clumped together, people understood the role of adhesion. They understood sort of later when we get these adhesion proteins that would, be responsible for this. And so you can actually understand this in terms of adhesion energies and differential adhesion energies across space. And this describes sort of how these get aggregated into specific patterns or randomly mixed cells. And there's like reference 48 and 49 here. I don't know what those are. But despite the self-organized nature of these processes, it is possible to make some predictions concerning the spatial arrangements at a steady state. So these are where you're programming assemblies of cells. You're dealing with the adhesion of these cells, and you're sculpting the multicellular milieu. And finally, synthetic morphology and genteel materials. So one way of moving beyond the cell level engineering involves considering cell collectives as genteel materials. And this is what Jesse's been talking about earlier in the meeting. These systems exhibit emergent properties at the system level that cannot be understood in terms of the properties of the constituents. So these are genes and cells. So this is where we're looking at mechanism, but at a higher level than genes or cells. We're looking at it at the material level or at the morphological level. So cells, the collective of cells, are the material. The material then has the level of agency, not the cells or the genes. So this is what they're getting at with agential materials. And so, this would be important if you're trying to develop, say, a smart material or some new material that you wanted to program the shape of, or the other physical properties like hardness or other things. So this is something that we definitely it kind of relates to this idea of mechanism. This approach takes advantage of higher order properties of embodied living matter, such as memory, context-sensitive navigation of problem spaces, and homeostasis, to perform computations and design morphologies beyond the bottom-up principles of synthetic biology. So this is where we're doing this bottom-up stuff in synthetic biology, and we're programming assemblies, we're building circuits, but then we have this agential material that concept that is sort of the top-down aspect of this. So in a lot of complex emergent systems, you have the bottom-up components that are sort of the the effects of lower levels like genes or cells, and they're contributing to sort of the shape of the collective. And then this sort of aspect of the material layer or the morphological layer constrains what those things do. So, this is where you get this sort of constraint on these things that are coming from the bottom up. So this is kind of, speaking to complexity theory, but also speaking to some of the things they're doing in multiple layers to engineer these materials and morphologies. This approach takes advantage of higher order properties of embodied living matter to perform computations and design morphologies beyond the bottom-up principles of synthetic biology. This class includes organoids and biobots and other multicellular assemblies capable of collective responses in space and time and novel forms of behavior. So if we look at some of these references, so 35 is, so you can go back to 35, that is, of course, this paper, Rewiring Cells, Synthetic Biology as a Tool to Interrogate the Organizational Principles of Living Systems. This is back from 2010. This is annual review of biophysics. So that's, if we look at the references here, then we can see that there are a lot of different references. In general, we have different references of multicellularity and evolution. This paper here, Synthetic Symmetry Breaking and Programmable Multicellular Structure Formation. This is from 2023 in Cell Systems. We have papers like this paper, The Origin of Metazoa, a Unicellular Perspective. This is where we're looking at the origin of animals as individual cells and kind of coming together. So yeast actually provide a really good example of this, where yeast are metazoans, but they're single cells. Sometimes they live in these multicellular aggregates. And then, of course, the common ancestor of yeast with us, those are obviously multicellular. And so multicellularity has come to sort of a refined, existence with metazoa. So some of these things, where we have the physiogenetic determinants in the evolution of development. This is from Newman in Science 2012. Dynamical patterning modules, a pattern language for development and evolution of multicellular form. This is a developmental biology paper. Some other papers from Bongard and Levin, scalable pipeline for designing reconfigurable organisms. This is a PNAS paper from 2020. This is Blackiston, Bongard, Levin, and Kriegman, biological robots, perspectives on an emerging interdisciplinary field. This is from Soft Robotics in 2023. And let's see, there's a paper on synthetic symmetry breaking in programmable multicellular structure formation. This is Cell Systems in 2023. This is Wofford et al. So you can see that there's a lot of prior work that this was based on, kind of going through the world of sort of synthetic biology, the evolution of multicellularity, and some of these papers on, developmental biology and developmental pattern formation, and then linking that to soft robots. So that's just kind of a cursory overview of that paper. I think it lays out sort of the what's involved in that field, what people are looking at, and sort of unifying sort of origins of multicellularity with some of these synthetic biology approaches, and really kind of getting at the question from a different angle than what a lot of people are interested in, which is kind of like defining what multicellularity is, and then looking and saying that, it's kind of maybe inevitable. And you see with synthetic biology, you have a lot of variants that maybe aren't obvious examples we see in evolution. So I think it's a good paper. I didn't want to get through all the details in it, but basically that's the take-home message, I think.
Yeah. Well, I mean, it certainly is the kind of thing we expect from Levin at this point.
Yeah.
Where it's like he will have a different take.
Yeah.
Where, it's not he will have a different take. He will bring you to see everything in a different way.
Yeah.
And yeah. I mean, it's a great paper. It speaks to that collective intelligence view of things. Right. That really is so profound and just permits so many different kinds of behaviors and needed kind of new fields to handle all the different ways that so I mean, this is great. I've been trying to watch more of James Glazer's talks. And and trying to think about, I mean, like I forget what he calls his class, but it's something like Introduction to it's like it's system biology, a network perspective, I think, or something like that, right? And just how there's already so much there, and that's really just kind of like that's mostly just like the ODE version of this.
Yeah.
Right. That like it's not really trying to get at some of this much more complicated stuff. Well, I don't want to say more complicated stuff. To get it's still not even at this perspective.
Right.
Although CompuCell 3D certainly has models that start to get at this. But I was just wondering about how like one, trying to think about what's the new biology kind of set of texts and classes.
Yeah.
and certainly James Glazer's like a start. Maybe this is more of systems biology kind of set of classes. There's a I posted this that one of his postdocs is producing a kind of multi-scale course.
Okay.
And I posted a link to it. I think they're calling it like embryo, like EMBRIO.
Oh, right. What channel is that?
Oh, I think I might have been on DevoWorm orthogonal, because it seemed kind of in line with some of the things.
Yeah. This is
things you cover there.
Embryo multi-scale modeling course.
Yeah. You know, and again, there's good stuff there. There's another kind of like biological or genetic circuits from Caltech. Anyway, the main thing being these are all interesting, but the other thing that I don't know if they talk about, and I'm curious when Levin will get to this, because you know he will, is like immunology.
Okay.
Right? Like how does immunology differ in this? You know? Because when they're talking about agential material, they are kind of talking about multicellular.
Yeah.
You know, and it's super interesting. Anyway, I was just thinking about James Glazer does a weekly course. Well, no, sorry, weekly seminar series. Thursdays, it's noon Pacific time, because I can't make it because I got another meeting.
Yeah.
And there's like 3 meetings that same hour. And he Galim print. I don't know what the acronym is. Anyway, that's their glimprint.org. Yeah, a global alliance for immune prediction and intervention. Okay, Glimprint. Anyway, it's just really interesting. You know, there's a lot of their work in terms of like digital twins with, you know, with immunology. Right, and just the kind of the weirdness of immunology.
Yeah.
You know, and you know, as if biology needed more complexity.
Yeah.
but wanted to mention it because he was saying as well that he's actually looking for help on things like just the website and stuff like that. And I was gonna kind of nose around a bit.
Okay.
But just also, you know, like as interesting as this is, it doesn't always get in the kind of the interesting things that the immune system does. And there was another interesting talk on, and I forget where this was from, but do microbes have immune systems?
Oh, okay.
And which made me think a little bit about the French paper on, you know, the you know, kind of brains in single-celled organisms.
Right.
You know, it's a similar thing, right? It's just like can a single-cell creature have an immune system? Just like can a single-cell creature have a brain? You know, which strikes you as a kind of multicellular creature.
Yeah. All right. Yeah, that's great. Well, thanks for the feedback, and that was a good paper. So yeah, next week, yeah, we'll keep up the conversation in Slack, and see you next week.
Take care.
Take care.